Hello everyone, my name is Federico Cerri and I'm a biology graduate student at the University of Milano Bicocca. I'm the first author and presenter of our research project about bioprospecting of Maldivian soft coral. Today I will present our study on chemical ecology and cytotoxicity of their secondary metabolites, focusing on the biochemical investigation of their effect against anti-cancer cell lines. Corals mostly belong to the Anthozoa class, which is divided into two subclasses, Hexacorallia including hard corals and Octocorallia including soft corals. Octocorallia have received more attention as potential source of drug candidates since, as you can see, they do not possess a calcium carbonate exoskeleton. And therefore they can survive in competitive environments thanks to their chemical defense strategy based on a series of secondary metabolites that accumulate in their tissues or are released into the surrounding environment. These compounds have been frequently investigated concerning their possible anti-cancer effect by testing their cytotoxicity against cancer cell lines. We have studied this aspect in our recently published review, that is Cytotoxic Compounds from Alchonide, an overview of the last 30 years. In this publication we focused on Alchonide, a large family of soft corals that includes three of the most common soft corals genera, Sarcophyton, Lobophytum and Sinularia. In this huge work, we reported 344 cytotoxic compounds with cytotoxic activity against different types of cancer cell lines, such as colorectal adenocarcinoma, breast carcinoma, hepatocellular carcinoma, acute lymphoblastic leukemia, and others. 70% of these compounds were isolated in the last 10 years. Proving that this is a newly explored research field which will grow further thanks to rapid improvements in analytical technologies. Besides confirming the potential of this research project, 21 of these secondary metabolites displayed comparable and even higher cytotoxic activity than the chemotherapy drugs used as positive control in the various studies against specific cancer lines. To start, we focused on Sarcophyton, an octocoral genus with a characteristic mushroom shape, as you can see in this slide. For our initial and preliminary experimental work, the first sampling of Sarcophyton was done with scuba diving at a depth of about 10 meters near Digura Island, located in the area atoll, as you can see in the green box, which is situated in the Maldives. The metabolites were extracted using two solvents of different polarities, ethyl acetate first and dichloromethane later. Starting from 65 grams of coral sample, we obtain 1.57 grams of ethyl acetate extract and 275 milligrams of dichloromethane extract. To evaluate their potential cytotoxicity, the two extracts were tested on two human colorectal adenocarcinoma cell lines using this one and this one, using the so-called MTT assay. It is a colorimetric assay in which the change in the viability of the tumor cells was evaluated 48 hours after the treatment with the two extracts. In the two graphs above, the treatment was done with the extract in ethyl acetate, while at the bottom the treatment was done with the extract in dichloromethane. It is evident that both extracts are cytotoxic on both cell lines. In fact, as we can see, for example from the first graph, as the extract concentration increases, once a certain threshold is exceeded, the viability drops dramatically until it approaches zero at the highest concentrations of the extract. To facilitate the comparability of different experiments, a unique numerical value is extrapolated from the dose response curves, these curves. This is IC50, which is the concentration that determines a reduction in viability of 50%. The lower this value is, the more cytotoxic the extract is. In this slide, we see what happens at the microscopic level. In these three plates, the same amount of cancer cells were seeded, about 1 million. After 48 hours, this is the situation without treatment, whereas you can see that the tumor cells proliferated until confluence. On the other hand, this is the situation 48 hours after treatment with the ethyl acetate extract in a concentration equal to the IC50 value. As you can see, the proliferation is significantly reduced. Besides, after 48 hours from the treatment with a concentration higher than the IC50 value, the proliferation is almost zero and most of the cells have gone to death. Below we see the same thing, but regarding the second cancer cell line. It was decided to purify the ethyl acetate extract by silica gel column chromatography, and the eluate was collected sequentially in these cylindrical glass tubes. Preliminary chemical analysis using thin layer chromatography made it possible to combine these glass tubes in 10 main fractions. It was decided to evaluate the possible cytotoxicity of 3 of these, the fractions 0, 2 and 5, which were the most different ones from a chemical point of view resulting from the preliminary analysis. These 3 fractions were further chemically characterized by gas chromatography mass spectrometry, GCMS, using the instrument shown in this slide. This is the analysis of fraction 2. Each peak in this graph corresponds to a molecule contained in the sample and detected by the instrument. The height of the peak is proportional to the amount of each molecule. Therefore, the higher the peak, the more abundant that molecule is in the sample. The GCMS also allows to associate possible corresponding molecule to each peak with a certain match. And the associated molecule is the one that has the highest match among those present in the instrument library. And this match derives from the correspondence between the mass spectrum of the molecule detected in the sample and that of the molecule present in the library. The analysis was done for all peaks, and it was found that this fraction was mainly composed of molecules belonging to the so-called class of terpenoids, steroids, and sterols. The same thing was done for fraction 5, which turned out to be lacking terpenes, but enriched in esters, alkanes, and alcohols. Furthermore, this analysis was conducted for the dichloromethane extract, which proved to be, as you can see, extremely heterogeneous. The most interesting note comes from the fraction 0, which turned out to be the purest fraction. In fact, as we can see from this graph, it contains a few compounds, of which 2 represent the vast majority, including this one, which corresponds to a wax known as acetyl palmitate. For the reason that it is a relatively pure fraction, it was possible to demonstrate the actual presence of this wax using a more advanced analytical technique that allows elucidating the structure at the atomic levels of the molecules contained in our sample. It is the nuclear magnetic resonance spectroscopy. This is the atomic structure of the molecule, and the hydrogen atoms of each group are associated with different peaks in the proton nuclear magnetic resonance spectrum. the carbon nuclear magnetic resonance spectroscopy instead detects the carbon atoms of the different groups of the atomic structure of the molecule, which are associated with different peaks in the spectrum, as we have seen in the previous slide for the proton spectrum. Cytotoxicity of the fraction 2 was evaluated by the MTT assay. In this graph, the orange and the gray lines are the two colorectal cancer cells, and as you can see, it is evident that the viability decreases as the fraction concentration increases, resulting in these low IC50 values. However, the interesting thing is that the effect on the normal cell line of the colon tissue, that is in blue in the graph, is much lower. In fact, as the fraction concentration increases, viability is maintained above 80% up to high concentrations. Therefore, this fraction is very promising as it is strongly cytotoxic against the tumor lines, but weakly active against the normal one. Fraction 5 is also more cytotoxic on tumor cell lines than against a normal one. However, as we can see from both the IC50 values and from the graph, this difference is less evident. This work represents only the beginning of our research project. First of all, we will purify the various fractions and isolate new single bioactive compounds. Once we have tested these compounds, we will try to investigate the characteristics common to the most cytotoxic molecules. But above all, we will try to define the cellular target, the mechanism, and the intracellular pathways involved. Finally, we will try to extend this study to other coral genera in order to identify the most promising ones.